Glad to see everybody here. And as Jill said, I'm the director of Biological Mass Spectrometry here at IU Bloomington. And we're a mass spectrometry primarily research laboratory. And as you'll see today, biological mass spectrometry usually involves protein analysis, although we'll touch a little bit on metabolites as well, and some glycosylation analysis, although not glycomics per se. As Jill mentioned, I want this more to be a conversation, so feel free to interrupt. I'll sort of be glancing over, but I don't know if I always pick up the chat messages popping up. So definitely unmute yourself and ask a question if you have any particular questions about a given slide. So, so let's just get started. Just a little bit of orientation. I know we're all spread out around the state, but the building on the right here is called Simon Hall. That's where we're located, right next to chemistry here on the left. But this is the kind of sciencey area of the campus, or one of the two sciencey areas. So right behind there is the biology, or several of the biology buildings. But Simon Hall is a sort of interdisciplinary research center. I'm based out of chemistry, but the building is filled with sort of chemists, biochemists, and cell biologists. So as I'm sure you're all aware, being able to walk down the hall and chat with your collaborators really improve the sort of bandwidth of your science. So today, what I wanted to do was give you a flavor for the types of things we do here, and in a more practical sense, some experimental parameters if you're a more biologist about how you might interface with mass spectrometry. And then I wanted to talk a little bit about my particular interest, which is protein glycosylation. So if I'm jumping around too much from topic to topic, I apologize, but I just want to really highlight some of the recent published work that we've done with various collaborators. So, so mass spectrometry, of course, is a really broad field, and the types of things that we do as a collaborative research service corps are also quite broad. And so some of it, in a way, is very, I mean, I wouldn't say trivial, but as the field progresses, certain basic things that we can do become commoditized, in the sense that there are fairly standard assays that you can get anywhere. They serve a really important purpose, right? Because people need to know their protein molecular weight, or identity of something that co-immunoprecipitated, but they aren't quote unquote research. They're more standard assays. So, but I'll give you a flavor of everything we do. So something simple like an intact molecular weight determination, or something more complex like native electrospray, where we try to preserve the three-dimensional structure of proteins during the electrospray process, so that we can look at larger protein complexes. The bread and butter of proteomics these days is identifications of proteins, from very simple to very complex mixtures. That's mainly done, I'm not sure I like the term, but bottom up. In other words, we don't analyze the proteins themselves. We digest them with a variety of proteases and identify the smaller peptide mixtures. The lab's also equipped to do various metabolomics assays. Quantitative analysis is really useful, right? You can just layer that on top of your protein characterizations, and depending on, of course, the assay, that will tell you a lot more about what's going on. In general, sort of my real research interest is post-translational modifications, and sort of as a corollary, looking at protein-protein crosslinks, either native links such as disulfide bonds, or more commonly chemical crosslinking to get some crude three-dimensional structure for protein complexes. All right. So we have a lot of protein biochemists on campus, and they're often just synthesizing recombinant proteins, and they just want to know the intact mass analysis. So standard LCMS, really simple gradient, and we deconvolute the data and get them the intact mass. It's a fairly accurate process, about 1 Dalton in 15,000 sort of error. If you ever ask a mass spectrometrist about a parameter, they're going to say it depends. And so they often ask, well, how much protein do I need? It really depends on, you know, the behavior of your protein and the purity. So this is sort of a very conservative estimate. But, you know, in a small, well-ionizing protein, we could use 1/100th as much. But I try to get people to give me about a half a microgram, which for a protein biochemist is sort of a rounding error on their synthesis. And for just these simple protein IDs, they can handle 1 or 2 or 3 different proteins in the mixture, or possibly more variants. So let's say you're looking at a post-translational modification on your protein. We can see multiple things in the raw data, but because these individual proteins give us so many peaks that we have to deconvolute, it quickly breaks down if we have a complex mixture. And here's an example of what that data might look like. I'm assuming you all have a sort of basic familiarity with mass spectrometry. I didn't want to go into too much sort of, you know, intro background here for purposes of time, but here is a protein that's coming off of an LC column. So it's being sprayed in acetonitrile water with some formic acid. It's going to be, quote, fully denatured, and all of the lysines and arginines are going to potentially pick up charge. And so for one given compound, this is a single protein, we see a range of charge states. You can see the spacing gets closer together. This is M/Z. So the same exact mass, but we see increasing protonation. So each of these adjacent peaks differ by 1 Dalton. And so if we zoom in, we can see the mass of each of those peaks, you know, is labeled. We don't see, for the cognoscenti in the audience, we don't see in this case the isotope envelope, right? So this particular protein, there might be 200 peaks under there representing various numbers, primarily of carbon-13 isotopes. And so we don't know formally the charge on any of these peaks, but we do know from our understanding of biology that or biochemistry, whatever the charge on this is 1 more protein and a proton, and this is 1 less proton. So, you know, the formulas look intimidating every time I show them to the grad students, but the concept's very simple, right? This mass times X, where X is the proton, is the same as this mass times the quantity X plus 1, and you just solve for the algebra. And, you know, obviously in practice, we have the software do the deconvolution, not because the math is particularly hard, but if you have a more complex spectra than I'm showing here, right, there's really one thing here. Maybe there's 5 PTM modified higher mass, you know, forms on the right side of this tail. We don't want to have to deconvolute all that ourselves, that have the software sort of simultaneously analyze it. All right, and so that's, again, one of the more basic things we do. Here's something more, you know, analytically interesting or challenging to us, which is some results from intact native electrospray. So let's say you have a protein dimer, trimer, multimer, and you want to, for whatever reason, spray it in its native form so that you can see the intact complex. Maybe you're trying to determine isotope, or subunit composition or stoichiometry. Maybe you're looking for a non-covalent cofactor that might be present, and that the standard LCMS that we do, which gives us very good data, has the downside of denaturing the protein. So you lose all that information. But we can just spray these things under gentler buffer conditions with more amenable electrospray voltages and temperatures, and get, in this case, a heterohexamer mass. And you can see, of course, the data is much less intense because the ionization efficiency of these things is, you know, an order of magnitude lower than that of the standard LCMS. But here's an example of a 162 kilodalton protein complex. Here's another example of a 121 kilodalton multimer. And so the biochemists in particular find this very useful, in a particular system. All right, so most of what we do though is peptide based. For our number of experimental reasons, it's much easier and in a way more informative to determine the subunit composition of peptides. And by peptides, I mean something from, let's say, 7 to 20 amino acids in length. We can generate those from a proteolytic digest of our sample, and we can use LCMS to sequence them, LCMS, and then map those back to protein structure or protein sequence. And these things can come from a range of samples. We're somewhat moving away from gel bands here at IUB. I mean, if it makes sense from your biological sample prep to do an SDS page gel, then by all means, you do one, but we tend to do more in solution digests. And if it works well, it's sort of easier for everyone involved. And these things can be as simple as IP proteins, or they can be whole cell lysates where you want large scale, on the order of 1 to 5,000 proteins quantified across sample replicates. All right. I basically have already made this point. The protease of interest, or that we typically use, is called trypsin. It cuts after arginines or lysines, which make it very useful for mass spectrometry because arginines and lysines have positive charges, and those positive charges are then localized to the carboxy terminus of the peptide. And because the mass spectrometer is really a mass to charge spectrometer, we really like to have charges on our peptides, in this case, multiple charges, so that we can basically profile all of the resulting fragments. Nevertheless, trypsin's not an ideal enzyme for all applications, and we often resort to a panel of other enzymes with varying specificities, such as chymotrypsin or aspen or pepsin, to really map out, in the case of individual projects that really want to focus on, let's say, one protein and all of its PTM states, we use a panel of enzymes to get there. And the enzymes, of course, are going to create peptides of varying lengths depending on the protein sequence. But from the mass spectrometer point of view, those between, let's say, 8 to 25 amino acids really give us the most interpretable MS2 spectra. And as I said, I don't want to go too much into basic mass spectrometry. I'm assuming you didn't really come here for that, but just to set the stage here, it's an example 4 amino acid residue peptide, just to show you what's going on schematically. So we typically use what's called collision-induced dissociation after we've taken an MS1 scan, identified everything that, let's say, is eluting from our LCMS system at that point, and then the instrument will iteratively select the most intense peptide precursors, isolate them one at a time in the instrument, and then subject them to vibrational fragmentation. When you do this, the weakest bonds in the polypeptide are the ones to break first. Those turn out to be the peptide bonds themselves. And keep in mind, you might have millions of copies of a given molecule or peptide. And when you do, and you're spraying them all together, some of them will break here, some of them will break here, some of them will break here, and you'll get what's typically referred to as a ladder of fragment ions of increasing mass. And it would look something like this. And so if we see ions from the carboxy terminal here, they'll go in the order of Y1, Y2. So Y1 would be a break here, it would be this part. Y2 would be this part. Y3 would be this part. And the nomenclature just has the ions from the amino terminus be B ions. So that would be B1, B2, and B3. And you look at the spectra, and if you sat there with a pencil and paper, and the table of all the amino acid residue masses, you can start to find pairs of masses and walk along to sequence the peptide de novo. Of course, we don't do that. We don't use that exact algorithm either by hand or by computer. What we end up doing is pattern matching against protein databases. So unlike gene sequencing, where you really don't need to know anything to read off the exact residue pattern, modern mass spectrometry or proteomics really relies on pre-existing protein databases. And so what we would do, here's an example of, for example, the Y-ion series that was matched by the algorithm. But what the algorithm really does is it takes the protein database, let's say this is human, 21,000 or so proteins, and we tell the machine or the computer what we digested with, so trypsin. So it creates an electronic copy of all the potential peptides that could have been generated from a tryptic digest of the human proteome. It ranks them by their theoretical mass. What's not shown here is the MS1 data used to trigger this MS2 acquisition, but from the MS1 data, we'll know the mass of the precursor, within a couple parts per million. We basically just then go to that list. We look at the, maybe 50 or so peptides that are close enough in theoretical mass to our observed mass. And we say, okay, these are our 50 candidates. And then because we know how the peptide is supposed to fragment in theory, we can generate theoretical spectra for all of them and do pattern matching between the actual observed spectra and the 50 or so theoretical spectra and figure out which one is the best match. Of course, you'll always have a best match. That doesn't mean it's right. So we do some secondary statistics to say how much better is this match than would be expected from the decoy distribution. And we get statistics on the confidence of our hits. All right. We do a lot of quantitative analysis here. There's a number of different approaches. For everyday use, I guess, as it were, we standardly output what's called label-free quantitation. So for all the peptides identified, the software can go back and figure out the MS1 level LC-MS intensity to give us not actually, the data is actually pretty good these days compared to earlier generation machines. So even if you don't do anything more than that, you can get pretty good quantitative comparisons across samples. Stable isotope labeling still has a lot of advantages. That originally was developed to, or one of the original flavors was metabolic incorporation. So let's say you're growing cells in culture, you can of course control the media. You would put in isotopically labeled amino acid precursors. Those would get incorporated into proteins and you can quantify that way. I think more commonly, it's hard to speak for the entire field, but more commonly, chemical labeling approaches have really come to the fore because they have a lot of advantages that we'll see in a second here, which is has to do with their multiplexing. So this is an example of something called a tandem mass tag. There are, it's put out by Thermo, but there are other companies who do similar sorts of technologies. And what the approach they've taken is they have a reactive group, this NHS ester. This reacts with primary amines. So after you've digested your sample, all of your amino termini of the peptides are able to be labeled, but as well the lysine side chains. So this sort of goes away, or this part falls off, and you get these two parts, the reporter group and then this mass balancing group. And you notice there's a lot of atoms here. There's a lot of carbons, there's a lot of nitrogens, and there's an oxygen. And you can use different combinations of isotopes such that you can make many versions. I think 16 is the number now. 16 versions of this tag that all have the same nominal mass. And so you could take 16 samples, label them each with a different version of the reagent. Here's some, I think these red asterisks, I know it's hard to see here, but indicate what atoms have isotopic incorporation. But you could take up to 16 samples, label them, mix them together, and the MS1 peak for each of those, for a given peptide, regardless of what version of the reagent is, it will have the same mass. And so those will all coalute when you want to fragment them. they will all be selected by the instrument together because the instrument isolates, let's say, a 2 Dalton window, and then they're all fragmented. Coming back to this, what they've done is the reporter group is of varying mass, and then this balance group is also of varying mass, and the two of them offset each other. So the reporter group falls off, you don't really see it, but you get a series of different reporter ions in the low mass region of the instrument. Oh, sorry, for the cognoscenta here, this is actually eye track. This is the TMT tag. You can see it's much bigger here. There's many more ions or many more possible isotope locations. Anyway, so in the MS2 itself, you get the sequencing information, but you also get the quantitative information. And the real benefit there is the large sample numbers that you can quantify in a single LCMS run. Here's just a recent example. These plots tend to look the same from these sorts of experiments. It's the so-called volcano plot, where the collaborators were looking at mutant protein line or a cell line with a mutant protein, and they wanted to see how the protein expression changed. So on the x-axis, log fold change, to the right of 0 is increased, to the left is decreased. And of course, if you have enough replicates to do statistics, you can plot out the log of the p-value on the right, and this will tell you, depending on the quadrant, what was statistically increased versus statistically decreased, in your sample. All right. We have ion mobility in the lab as well, in the form of a Synap G2S, which is an instrument from Waters Corporation. I don't want to get into the details too much, but this gives you further enhancement of label-free quantitation ability. So just as a sort of large or medium large scale experiment, we looked at 50 different Drosophila samples from, in this case, it was a time course of age of the Drosophila, and could quantify around 1,200 different proteins. We're just plotting them out based on some gene ontology groupings. And you can see it's a log of abundance. So we're getting about 3 orders of magnitude of scale here, which of course is important because, again, depending on the system, but let's just say in humans, I think the number that people kick around is more like 6 orders of magnitude of differential protein expression between, something like a histone or an actin protein that's millions of copies per cell versus something at low abundance like a transcription factor. So, in full disclosure, right, proteomics is not where genomics is in terms of the depth of sequencing coverage, but the field and the instrumentation has certainly made really good progress over the last 5 or so years. All right, give you an example of the metabolomics that we do. I should say there's another mass spec facility here that is based in chemistry itself, and they tend to deal more with small molecules. So metabolites is sort of an interface where we both do some work depending on, whose instruments are a little more tailored for that sort of particular analysis. But just as a straightforward example, collaboration with Greg Demas in biology here, they wanted to look at a range of steroid compounds they've done multiple experiments over the years, but as a function of light dark cycle as a proxy for sort of seasonality. So looking at how steroid compounds vary in the blood or the brain of rodents and how that tracks with aggression. And so we've set up assays. These are, again, if you're a mass spectrometrist, you might expect these typically to be done in a triple quadrupole instrument, which is a type of mass spectrometer that's really designed to do nothing but quantify compounds of known interest. We're sort of playing around. We actually did these experiments on an Orbitrap where we would sort of get the selectivity, the SRM aspect of the quantification by doing high resolution extraction of MS2 fragment ions measured out in the Orbitrap analyzer. So these types of quantitative assays are often very sensitive because you have two levels of mass selection. And so even though many small molecules might have similar-ish masses, since you're also measuring the mass of resulting fragment ions, the odds that two compounds have both a similar precursor mass and also generate certain similar massed fragment ions is pretty low. So this is sort of an ongoing series of projects. Now I want to transition over to post-translational modifications. There's sort of two ways that this plays out experimentally, at least in terms of like what collaborators want. Very often people have a protein of interest, and for, let's say, because it's running aberrantly on a page gel, or because they're treating it with a kinase in vitro, or any number of reasons, they may believe that it's post-translationally modified, and so they'll give us some of this, and then we try to identify what's going on. If that's the case, then the parameters that are really important aren't so much our ability to enrich a given PTM, because they've really just given us a purified protein at that point. And the challenge is getting sufficient amounts of protein. When the person, again, is sort of a biochemist, when they're recombinantly expressing it in some heterologous expression system that's really been designed to give you lots of protein, then they can give us lots of protein, and people tend to be happier. In terms of we can find more things the more sample you give us, right? But of course, in the real world, or in parts of the real world, somebody might want to immunoprecipitate endogenous levels of their protein of interest. And then this presents, of course, much more of a challenge at our end. Not that we're doing anything particularly differently, but we just don't have that much material to work with. All right. Are there any questions at this point? Nope. All right, I'll continue. So I'll talk a little bit about cross-linking first, and then get to post-translational modifications. But protein-protein cross-linking, has a lot of obvious applications. In particular, when you have a multi-protein complex and you want to be able to, roughly map protein-protein interfaces. Obviously, if you could get a crystal structure or a cryo-EM of this, your protein complex really easily, that might be, a higher resolution sort of data set. But lots of collaborators come to us sort of intermediate to having samples that will be amenable for those additional approaches, or just, it just doesn't work for those other approaches. So there's a lot, if you look at chemical catalogs, there's a lot of different cross-linkers out there. In our experience, NHS esters are the most sort of robust chemistry. So an NHS ester would be this N-hydroxy-6-cinnamidal ester, and you would get a bifunctional cross-linker with two NHS esters on either end. And then, maybe you throw in some small molecules to make it a little more water soluble. Here's an example that I think has kind of recently come out. We've had some good data with this cross-linkers that are able to be fragmented in the mass spectrometer. Anyway, you can vary the length of these, but these NHS esters that react with amines work in our hands, or in the literature as well, I would say, much better than 99% of other types of cross-linkers, no matter how attractive they would be in theory. I should also give a little plug for CDI, which does cross-link amines with carboxylic acids. And we have had some good experience with that as an alternative. So here's an example of what a cross-linked spectra would look like. So here's one sequence in green and the other sequence in blue, and you can see the corresponding labeling of different ion types. This, of course, since I'm showing it to you, is a really good example of a data-rich cross-linked MS2 spectra. Somewhat anecdotally, what we've noticed is a tendency when we have cross-linked peptides for one of the peptides to give us really nice fragmentation, a really nice fragmentation pattern at the expense of the fragmentation pattern being intense for the other half of the crosslink. And that's obviously unsatisfactory. Knowing half of a crosslink doesn't really get you where you want to be. And again, this is somewhat anecdotal. We haven't, done rigorous quantitative comparisons, but that's why we've been pretty excited recently about this DSBU version of these NHS esters. So DSS/BS3 are essentially the same thing in terms of the crosslink they make. And I would guess from the literature, this might account for sort of 75% of what's published out there. And this is a somewhat newer version, but the fact that it will also break apart during MS2, in my experience, seems to indicate that when we get secondary fragments, we basically separate the crosslink, and then the machine is able to fragment the two halves because they're separated now independently. While they're still stuck together, just the way that collision-induced dissociation works, it's going to be biased towards the weaker bonds, but I think it's also going to be biased to the bigger structure. And so we've gotten what I think is more evenly distributed fragmentation from these gas phase cleavable reagents. And so then what do you do with this sort of information? Again, what you can do is, let's say here's a multi-protein subunit, and they're wanting to know the interaction between POLB1 and PBP2. And so, this sort of figure, of course, is not at the level of, what cryo-EM might give you if you had the intact complex. But if that's not working for you, you can at least figure out what parts of each protein domain are interacting with their neighboring subunits. As I said, in addition to chemical cross-linking, we can also profile cysteine disulfide. So here's an example. I think it's not obvious here, but we were all actually using a multi-protease approach because the resulting cross-linked disulfide peptides were too big to give us good sequence. So so we would process our samples as normal. Of course, we're not presumably running an SDS page gel if those are, or at least a reducing gel. But you could take a protein complex. We normally reduce and alkylate cysteine disulfides for technical reasons so that they don't get in our way. But we can, of course, leave that step off and just digest the protein as is. Tweak our LCMS conditions so that we're trying to target larger compounds at higher charge states and find these disulfide modified peptides. All right, so post-translational modifications, I mean, there's hundreds of them. We typically talk about the obvious ones like phosphorylation, glycosylation, acetylation, and so forth. But this is a small cut from a larger figure that shows most side chains are modified one way or the other. At least if you look in the textbooks, right? We don't see these some of these modifications very frequently in mass spectrometry. In fact, we don't look for most of these more obscure modifications. Not the ones I'm showing, but the hundreds of others I'm referring to. We don't even look for them in our standard database search algorithms. And so we tend not to find them. There's good reasons from a statistical point of view not to look for things that probably aren't there, but that's pretty much where the field is. But if you want to find a post-translational modification, you could do it, as I said, from a purified protein, but perhaps more interestingly, you want to look more broadly at your cell type of interest or your organ of interest. And so because the proteome, especially after digested, is so complex, you really need specific biochemical approaches to enrich for your target molecules of interest. And so we've done a lot of work traditionally with phosphorylation using immobilized metal affinity chromatography, which will specifically chelate phosphorylated groups. We can purify those away from non-phosphorylated components and do standard LC-MS analyses, but of course, our samples are highly enriched for molecules of interest. So that would be an example of a phosphorylated peptide, and we can, of course, map what residue is modified if we have a good enough sequence. We've done some work with tyrosine sulfation that has a mass very, very similar to that of phosphate. What we're demonstrating here is that using a variety of fragmentation types, so standard collision-induced dissociation, a higher energy version of collision-induced dissociation, something called multi-stage activation, where we try to fragment both the precursor compound and intense fragment ions in the same scan. We can do that. We also have the ability to do what's called electron transfer dissociation, which often keeps labile modifications on the peptide. And so the take-home message of this slide, we don't have to look at every panel, but the phosphorylated version of this synthetic peptide has a fairly distinct MS2 pattern compared to the sulfated version of that peptide. All right, here's an example of some chemistry we helped develop with David Giedrock here at IUB. They were interested in persulfadated cysteine residues. So, normally you think of free cysteine SH or cysteines in a disulfide SSH, but there's a lot of free thiols floating around, and some high percentage of your cysteine residues actually are SSH. And so we wanted to be able to specifically pull this compound or this class of compounds out from a complex digest that includes this, and of course, all the cysteines that don't, all the peptides that don't have cysteine. So, here's what we developed, an approach using iodoacetamide linked biotin. So iodoacetamide is just a small molecule that in its regular form, pure iodoacetamide, we would use to cap free cysteine residues. And so if we don't selectively reduce our disulfides prior to digestion, and we just throw this reagent on, we'll label both the free cysteines and the persulfadated cysteines. And then because we have a biotin tag, we can enrich these two compounds away from this guy and the 99% excess or 90% excess proteins that don't have any cysteines on them. And because this has still a disulfide on it, we can then selectively reduce this disulfide, keeping this one on the resin. We elute, we then also want to cap this and then do LC-MS. So everything we see that survives this process that has carbamidylmethylated cysteine on it, because it's been then treated with regular iodoacetamide, was a persulfadated cysteine. And we identified on the order of around 500 different persulfide modified sites in Staph in the proteome. And here's an example where we're doing some sort of semi-quantification. The circle size, it represents individual proteins. The size of the circle represents the relative abundance based on label-free analysis of the proteome in general. And what we're plotting out here on the y-axis is the observation of MS2 sequencing events. It's a ratio of when we treated with sulfide stress versus not. So these would be the types of sites that have increased abundance in sulfide stress, and then these would be more standard state housekeeping levels. All right, so moving on to some glycosylation. glycosylation, we could talk for hundreds of hours about glycosylations. But very quickly, right, there's essentially, depending how you count, 3 types of glycosylation. This is an example of N-link glycosylation. In this case, the N refers to nitrogen on asparagine residues. So these types of structures get put on asparagines on extracellular or secreted proteins. Here's an example of O-type glycosylation. So put on oxygens of serines and threonines, also on extracellular regions and on secreted proteins. And then there's a third type of glycosylation called O-glycinac. So N-acetylglucosamine. This is a sort of a completely different mechanism and biology. It's put on and removed by enzymes cytosolically. So cytosolic proteins might be O-glycinac modified, but they are not only secreted extracellular proteins have N or O-linked glycosylation. And so we're particularly interested in N-linked. As I said, everything's about enrichment if you want to do a global PTM analysis. And so what we've developed is a way to purify glycoproteins or glycopeptides from complex mixtures using a class of proteins known as lectins. A lectin is really just a generic term for a carbohydrate binding protein. So the typical source of these is from plants, and individual plants have lectins that bind different types of glycans. So some might bind mannose, some might bind N-acetylglucosamine, some might bind sialic acid. And we can take either individual lectins or a pool of lectins, immobilize them on a resin and run it through a column and separate non-glycosylated from glycosylated peptides. Here's an example of the UV trace of that. And I'm just highlighting the region where when we look by LCMS, we see glycopeptides. You can do some kind of cool things with this. Normally, we use this as an enrichment approach and then do LCMS on the side, but you can hook a column like this directly up to LCMS if you use the right sort of buffers. And what you can see is these individual blue lines are the elution behavior of different glycopeptides in our sample. And these red lines are the elution behavior of different non-glycopeptides. And so because we're measuring them directly in real time, or as they elute from the column, we can really very tightly, of course, map their elution behavior, which allows us to test, in this case, sort of different resins or different buffer conditions to see what would optimize the separation of sort of the blue population from the red population. But what you can also do with this, and not just use it as a tool to optimize your enrichment, but you can then, by relating this to the retention time of known standards, you can actually directly calculate you can calculate relative affinities of individual sugar structures with individual lectins. And so these are 5 different lectins. We had immobilized on 5 different columns. We ran a series of standard glycoproteins that were digested, and we looked at how the sugar structures on those peptides on those proteins eluted from the columns. And so we can talk about how better map the exact affinities of individual lectins. All right. Going back to a sort of more biological context, we sort of scaled this up. Here's this column I talked about, and we wanted this in a more automated way. So we've attached a trap column. If this is the LC, that right as we get to this point in the elution, we switch the flow off of this column. Instead of going to waste or to be collected as a flow through, we now trap all of this. And that's important for 2 reasons. One is we have limited binding capacity in this column, so we can then scale up 10 times, let's say, run it 10 times, keep trapping on this same trap column all 10 tails that are glyco-enriched. Then we take this offline and we connect this C18 to an analytical column, and we just run LCMS, and then that allows us to collect fractions that can then be, this is an offline instrument. We collect the fractions, we dry them down, we inject them by LCMS, and we can find sort of tens of thousands of glycopeptides now. I want to talk a little bit about what we're doing with these glycopeptides, or what we're doing with the glycopeptide data. My sort of medium temporal background was in large scale phosphorylation, and that was great. We could get these data sets. We could get data, temporally as a function of stimulation or drug treatment. But it was really hard to connect the large scale nature of the data back to some biology, right? You could look to see the time course of things and group them by who's early, who's medium, and who's late in terms of response to your stimulation. And maybe that correlates with sort of the underlying kinases that might be active. But you get to that point and you're kind of stuck. And at least from my perspective as a sort of mass spectrometrist proteomicist, I think the glycosylation data is a lot more information rich. And that has to do with how you can think about the data and the relationships between individual glycoforms. I haven't gone into the nitty gritty of N-glycosylation, and this graph or this cartoon here is maybe a little hard to see, but this is the processing steps of one given glycoform as it goes through the ER, the cis-medial, and trans-Golgi. And the short answer is you stick on this very large sugar structure initially, and there's a series of well understood enzymatic steps that chew the branching pattern on this sugar tree down, and then start building it up with different types of glycans. So originally, these are all it's called high mannose, and then you start putting back on galnac or galactose or sialic acid or fucose. And then you maybe will end up here. And so what we observe from our proteomic data oftentimes is something called microheterogeneity. So for a given protein or a given peptide that has N-glycosylation on it, we'll observe 10 or 15 different forms of the glycans, some more abundant than others, but certainly more than one. And that's not really unexpected if you think about it, right? Each of these steps is a real enzyme that has less than 100% efficiency. And so if I found this in my sample, then logically I must have some amount of all of these intermediate steps, although they might be, orders of magnitude less abundant, but they should be there. But what we've taken to doing is using this data and plotting it out in a relational fashion. So, I could just give you a table of, 500 glycosylation sites I found in a sample, and, maybe there's 10 sites or 10 glycans per average on any site. I, got 5,000 rows on this table, and I can give you some quantitation information, but, what do you do with that? So we've tried plotting it out, and the trick or the tricky nature is that there's four main different types of sugars. There's hexoses, which might be mannose or galactose or glucose. There's hexnacs, which might be hexoses or there's hexnacs, which might be glicnac or galnac. There's sialic acids, which is usually just something called nu-GC, and then there's fucose. And you can have different numbers of each of these classes. And so you've got these 4 dimensions of data: hexose, fucose, sialic acid, hexnac. And we want to plot it out on a 2D graph. And so what we've taken to do is sort of embedding the information about what the sugar is by, in the integers in the x-axis here are the number of hexoses, and then we take the number of fucose residues we find and we divide it by 5. So something at exactly 6 in the x-axis has 6 hexoses and no fucoses. Something at 6.2 has 6 hexoses and 1 fucose. And we did the same thing for the hexnac. And so when you do that, you can plot out this thing I'm plotting out here in figure B is basically the same pathway as in figure A. It's just oriented sort of spatially with respect to the subunit composition. And I'm really just focusing on things that don't have sialic acid or NUAC or sialic acid or fucose here. You can see that here, which is a blow up of essentially one of these unit squares. So at exactly 5 and 4, it's got 5 hexoses, 4 hexnacs, no sialic acids and no fucoses, but you go over or up in 0.2 digit increments and you can express this. All right, and so I'm plotting out some other biosynthetic maps here, and then showing you some sort of nomenclature of N-glycans, where they might be in the map. But if this is just a map, it's not the kind of cool application. The cool application would be then plotting out the quantitative data, which we've done here. So each of these dots corresponds to a given glycoform on that particular peptide. So here's just an example of a protein with 5 different glycosylation sites. Each of these dots represent a glycoform we identified and quantified, plotted out in relation to, its biosynthetic neighbors. And so you can quickly see here, and I've color coded in red everything with a fucose. So you can see that this particular peptide seems to be about 50-50 fucosylated versus not, whereas there's a lot more fucosylation here. You can see where it is in the overall biosynthetic pathway. And this, I don't have time to get into, but this raises a lot of interesting questions about sort of mapping metabolic rates and thinking about the data, again, more in a relational way about how is this protein being actually processed as it goes through the ER and the Golgi. Some other things we do is a little bit of ion mobility mass spectrometry. This is a little bit more niche, but it allows us to separate things that can't be separated by a mass in the mass spectrum dimension alone. So here's an example of a glycosylated peptide. This yellow circle is meant to represent a galactose residue. We know from biochemistry that the galactose is bound to either this GLICNAC or this GLICNAC, but we don't know which it is from the mass spec perspective because they're the same mass. And using an ion mobility chamber, which is essentially, in a nutshell, a gas phase sort of SDS cage gel. So we can separate things based on how easily they move through a gas field, because the more compact they are, the more easily they'll move through. And we can see how they move through as a function of time. And so we see for this particular glycosylated peptide, for example, there are 2 peaks consistent with, of course, this being some of it being here and some of it being here. At this stage, we don't yet know which peak is which, but we have evidence that there are 2 forms of this sugar. Similarly here, there should be 3 forms at, maybe we could see 2 clear peaks. But we started using ion mobility to map out the linkage or how sialic acids, in this case, in particular NUAC, are linked to the galactose residues by fragmenting them and looking at the IMS resolution of the different pieces we see. This is something very similar, except we're fragmenting ion masses and trying to build back whether we have isomer 1 or isomer 2, or to what extent we have those 2 glycan isomers by looking at unique fragment ions that would occur in one form but not the other. All right, and so conclusions, perspectives, we can do a lot of stuff. We love to collaborate with people. We love more challenging experiments, and we love helping you out with your grant applications, or getting you even better to be so enamored with mass spectrometry that you make the proteomics formally part of your grant. And just to thank some people that have been shown up in these slides. These are former postdocs in the lab, and then Steve Bell, David Giedrock, Greg Dimas / Virginia Vitzthum, David Clemmer, and Charles Dan have been some of our more heavy collaborators over the last couple of years. All right, and there's some questions in the chat. I'll get to those, or you can. All right. Do we provide service for N-glycosylation profiling? So we've done this more as well, service versus collaboration, sort of a continuum. But if you want to drop me an email, we can talk. I mean, it's somewhat involved, as you might imagine. So it's not, quote, plug and play. But yes, we'd be happy to do a collaborative glycosylation analysis. Surface plus monoresonance, we do not do that. That would be like a BioCore instrument. I don't actually know all the brands of the surface plasmon resonance, but there's got to be a number of biochemical analysis facilities amongst the 3 universities or that do that, but we do not.
If anyone would like to unmute yourselves, please go ahead and ask your question. There's another one in the chat if you see it.
So again, so this sounds like, well, I'm thinking of like antibody biologics, right? So this would be a fairly straightforward experiment if I understand it correctly, right? For example, pharma companies, right, if they're going to make an antibody drug, need to do all sorts of QC on those compounds to validate that batch-to-batch samples don't change, for example, their glycosylation pattern. And so no one has asked us to map glycosylation patterns on an antibody, but I mean, the protein's sort of irrelevant, right? We digest the sample up, we generate peptides, and we look at them directly. And so, the analogy is we just, we're currently working on some recombinant spike protein from COVID, and we're just identifying and quantifying the relative glycosylation patterns on all of the N-glycans we see there. So that would be a fairly analogous experiment. So the answer would be yes, with the caveat that if it is what I think it is, which is the sort of assay that has been really, really, really well developed by pharma because they need it in a very, particular way to satisfy the FDA. I mean, I don't know who this question's from, but obviously that's not what you need, but you're not satisfying the FDA. But to the extent that it's a really well worked out assay with its own particularness, I'd have to go and read up on exactly what they try to achieve in those assays, in addition to sort of glycosylation characterization, just to make sure that I'm answering the question correctly. But can you give me a protein, whatever it is, if it's relatively pure and you have a couple micrograms of it, and I can give you an N-glycosylation pattern in a fairly straightforward manner? The answer to that is yes.
The question is from me.
Okay.
So the purpose is that a different antibody response. There's different phase of antibody response. There's early antibody response. That's most of the antibodies secreted are not or limited glycosylation. But when it comes to later phase, for some reason, antibody began to get glycosylated, especially the memory phase. For example, the glycogen-induced long-term memory antibodies are kind of more glycosylated. I just wonder if we collect the serum and there is a way to analyze the glycosylation.
Okay. So
like early versus late response.
Sure. So you don't have a purified antibody. You want to look kind of generically. Well, I mean, at the very least, right, you'd need some sort of protein AG, let's say, enrichment for antibodies, because otherwise, if we just digest serum, we're going to look at serum glycosylation. You're particularly interested in, broadly speaking, antibody glycosylation in serum at different biological time points. So if you could, isolate the antibodies from the non-antibodies, then it starts to become doable. I'm not an antibody expert, but the caveat with the experiment at that point is you still might have hundreds of proteins, right? I mean, you're at the very least, I don't know what you're, let's say you're inducing an immune response, right? With an antigen, you're going to generate polyclonal antibodies. And some of the sequences on the polyclonal antibodies will be the same and some won't. And, and there'll be IgG and IgM and so forth. And so if, but we can look at complex samples. So, but the one thing you might want to think about there is a glycomics approach. And we don't do glycomics, but other people do, right? So in glycomics, you formally cut off all of those glycans. So first you need to enrich your antibodies, then cut off all your glycans with an enzyme called PNGase F or something similar, and then get a glycomics profile by doing usually something like LCMS on the glycans themselves. And then you'll see changes. You won't relate them back to individual proteins, but it's not clear that's what you want. If you did definitely want to know, and I'm just throwing out IgG versus IgM, if you really wanted to know how IgG was changing, irrespective of how IgM was changing and vice versa, then yes, you would need to do glycoproteomics, not glycomics. And so then maybe that we would be the place to go.
Yep, thank you.
All right, and then changes in proteome within identified cell types as a function of experimental treatment. Yeah, so I mean, more recently we've sort of worked more with microorganisms for that, and so not that again, microorganisms might have their own quirks with breaking up the cell and solubilizing the proteins, but once we get to the peptide stage, a peptide is a peptide from our perspective, and so we can port those very easily. I'm only giving you some context for what we've done recently because any lab, us or anyone else, you have a certain amount of in-house resident memory. And so I've certainly done a lot more mammalian type cells in the past, but more recently we've done things with more limited proteomes. So we are shooting around the number 1,000 in terms of sort of protein IDs from sample to sample. And we're getting relatively good quantitative reproducibility. So yes, all right. Proteome analysis, either analyzing cell lysates or SDS gel bands. Oh, sorry, this is the same. Yeah, I mean, if you wanted a large scale proteome analysis, right? You don't want to fraction, if you do it in a label free, you want to make sure you have the same exact starting material each time. So in solution cell digestions of cell lysates is the best way to sort of preserve that. The problem with SDS page gel bands is, of course, running the SDS page gel the same way. Certainly if you want to cut out a region, at 2% change in drift of your protein down that band is going to really change, the comparison from gel slice to gel slice. We, for a while, what we were recommending to people on a related note is oftentimes people would bring us samples with detergents around, and detergents are very, incompatible with mass spectrometry. And rather than have people come up with a brand new way of isolating their sample, tailored to the mass spectrometry, because assuming that they've been working on this project for a while and they've got a good way to extract their protein, we tried to work with what they have and modify our analysis. So what I would have people do is run what I would call an SDS gel plug. So rather than run a whole gel, take a protein lysate, run it for 15 minutes or so, into the separating region. It might be, 1 centimeter, half a centimeter wide, and then cut the whole thing out. And the reason you would do that is it gives you an easy way to get rid of the detergent because your protein's immobilized in the gel. Of course, there's SDS around, but you can wash SDS and any detergent off. And then we do an in-solution digest, which is the pretty standard way to go about it. And that can be done. Again, we were really trying to get rid of detergents. We have ways of doing that in solution now that I think are better than they used to be. So we've sort of switched in that direction. But we certainly can do the sort of bulk gel band approach. All right. I had a question regarding cross-linking. I will unmute myself. Okay.
Hello, thank you very much for your talk. So I have been trying to characterize these vesicles that exist in the human plasma, and they are typically in the range of 30 nanometer to 100 nanometer. And
So
we would
exosomes?
exosomes, exomeres, supermeres,
Okay.
all these vesicles, even lipoproteins and VLDL, but we know enough about them anyway. So the thing is, with these newer vesicles, especially supermeres and exomeres, we don't know the complete proteomic profile that's present on the surface, especially which proteins are co-localized together. So I was thinking a cross-linking strategy would be efficient to identify which protein co-localizes with another proteins, because there are hundreds of proteins present on exomeres, and we want to do that because then we can develop some immunoseparation technique based on which are co-localized together.
Okay.
So the thing that I was worried about is that since we need relatively purer sample, the second we isolate these from the other constituents of human plasma, say albumin, these particles become very unstable. And so even once I have separated them out, they will start breaking apart, releasing their proteins. So if I, for example, do my crosslink, how do I know that it'll crosslink only the proteins on the surface and not the proteins that are present in the solubilized free-floating form? Thank you.
Well, so what you mean is you have a population, just to rephrase your question, you have a population of vesicles, some of which you think have broken apart and released the cytosolic contents.
Yes.
And so you have surface vesicles and then free-floating protein. Okay. First off, this would be a very difficult experiment to get good mass spec data on, because when we do cross-linking, typically what's worked well is having, protein A and protein B, and we cross-link them. It's all about enrichment for finding low-level targeted things. And when you have protein A and B, you've already enriched because, you don't have them in the context of the cell, which has 10,000 other proteins. And so we don't have to do any further enrichment. If instead, this isn't the experiment you talked about, but to take it to its extreme, you took blood and said, I want to cross-link proteins in blood. You can cross-link them, of course, but finding the particular cross-linked peptides after the digest in relatively enough amount to sequence by LCMS is going to be the challenge. So we could talk about this more if you want to send me an email, but there are other groups who have developed tagged crosslinks that allow you to enrich the cross-linked product itself. And so you're in a more intermediate state. I mean, vesicles. So we've done actually a fair bit of proteomics on exosomes, right? But I mean, obviously you're looking at multiple classes, but exosomes themselves we might find 500 proteins in them. Maybe some of those are impurities, maybe not. And so if that's where you are and you are prepared to biochemically beat on it hard enough with increased sample amounts and increased LCMS time, you could probably find in the mixture of digests some cross-linked peptides. Because the issue is, and this is why I think your cytosol is not going to be a problem, is, you got a random protein that used to be in an exosome floating at a reasonable concentration in your tube that has exosomes that haven't broken apart, right? The small molecule NHS ester is going to bump up to that and say, oh, it's got some lysines, I'll react with it, right? And now at this point, you have your protein, a cross-link, and then a free NHS ester at the other end of the cross-link. But because it's a cytosolic protein, let's say, that is not truly associated with anything, then there's no specific target. Now, it might be a cytosolic dimer and it's still intact with its dimer, in which case you'll cross-link to its dimer. But if that's not the case, you will eventually either hydrolyze your tag, because the NHS ester is hydrolyzed, or you'll randomly bump into something and cross-link something that shouldn't be cross-linked, right? By shouldn't be cross-linked, I mean isn't biologically close together. And that's maybe problematic, but because it's random, it's going to happen at super low probability. So you're never going to build up enough of that particular wrong cross-link, I would think, to ever see it by LCMS. Because you got a million other copies of this one protein, they're all going to bump into an NHS ester, and then they're all going to hit another random protein. And so you might, instead of having, the necessary 1 million copies of a given crosslink, you might have 100 copies of 10,000 different crosslinks, right? And so they're there, but you can't see them by mass spec. So the problem I think you'll have is actually seeing crosslinks in the first place, not trying to decide which of your crosslinks were randomly associated. That being said, you can quite obviously then go and look at, gene ontology, uniprot annotation of all of these things and say, why the heck is an extracellular domain cross-linking with a, cytosolid protein? Clearly that's telling me my assay's got some false positives that guy, John, who gave that lecture was confident wouldn't happen, right? So the data will tell you if I'm wrong, but I think it would be very unlikely to be a major issue. All right. Did you have any follow-up?
No, I think I'll think about it.
Okay, but yeah, if you want to learn more about sort of the tagged cross-linkers, you can drop me an email. All right, the next question: how to prepare detergent-resistant cell membrane proteins for else? So I guess are you still there? If you could just chime in, what exactly do you want done for MSMS? This is Kigui.
Hi, thank you for your nice presentation. So we are very interested to study virus receptors on the cell surface. So like smallpox virus, right? So we know we have vaccine to eradicate the smallpox, but so far nobody knows the receptors for this virus.
Okay.
So what we found is we did some mass spectrum to look at it, but when we extract the protein from the cell surface, and we basically use the soluble protein for the mass spectrum, right? But later on, we found when we immunized the mice with a soluble form of the protein was memory associated detergent resistant part. Actually, the antibody against the detergent resistant part blocks the virus binding. Okay. So therefore, these give us some idea the receptor actually associated with detergent resistant. We don't know whether it's a lipid or whatever that's actually resistant to the detergent. So we want to analyze that part.
Okay, so let me rephrase just so I understand, because I'm not an expert on the cell biology of membranes, but obviously there are different domains within the cell. And it sounds like what you're saying is you have reason to think there's a transmembrane, let's say, transmembrane protein in a part of the cell that's, of course, biologically important to you. But when you say detergent resistant, you mean when you lyse the cell with SDS or, RIPA buffer, you think it's not becoming solubilized?
Yeah, we lysis not use SDS, we use a tritone excited.
Right, John, yeah.
Right.
But I mean, I guess first thing I would say is, well, I would need to look up, how people biochemically, mass spec or otherwise, how they have biochemically characterized, quote, detergent resistant cell membrane proteins, because I'm not even sure how are you, because you could take the cell. I mean, there's always harsher things, right? 8 molar urea with 1% SDS is pretty much going to dissolve everything, I would think. You could imagine something crashing out of that solution, right? So it's still tricky, but you could take that. I mean, maybe not with all that urea, but you could take it and just run it on a gel, right? Or can you not? You don't think there's any way to lyse your cells and run it on a gel?
Yeah, we tried several type of the detergents, but when you run in the gel, you can see part of this protein actually stuck on the top. Never goes down to the lane.
So, if you just want to know the identity of it at that point, right? And I'm just kind of talking off the cuff here. I'd have to think about it a little more. But if you told me, look, I have this stuff, it's super insoluble. I can, the again, I always think of how do you isolate it? But you just told me you can isolate it by taking a regular detergent extract, running it, all the well-behaved proteins go into the gel, your protein doesn't. You could then take that stacker and you could try to proteolytically digest it, right? Because it's so poorly behaved, I'm not sure how well that will work, but you could also, you know, you could do things like cyanogen bromide, right? That is a chemical cleaver that breaks peptides at methionine bonds. And that would, I mean, after you've run the gel, your stuff is stuck at the top, you do some sort of chemical treatment to break it up a little bit, right? Because at the end of the day, if you just want the identity, you can be a little abusive to the protein as long as you could still get some tryptic peptides out of it at the end. So maybe maybe some sort of harsh proteolytic, there's something like called pronase or pepsin, these enzymes or enzyme mixtures that really like to chew things up. If you think you have a relatively limited number of proteins there, you can use some sort of very aggressive non-specific or semi-specific protease to generate peptides and still identify them. At the very least, you could see whether it chews it up visibly. I mean, maybe you can, in the same way that back in the day, like 2D gels, you would kind of do your separation and then lay your isoelectric focusing well into an SDS page gel. You could take your little insoluble bit, treat it with various things, and then just put that gel bit back into a well of a new gel and try running it and see whether your protein or bits of your protein will now run or are gone, right? And then you say, okay, well, look, I have a handle to start breaking this thing apart. I just want to break it apart gently enough to then do proteomics on it. That might be kind of my take.
Thank you so much.
Sure.
John, I think there was one question that we missed is, do you provide service for identifying unknown ligands?
Ah, yes. So, in theory. So we've had a number of people come to us and say, I have a protein, for example, I remember the details of one of them. I have a protein, I've crystallized it, I have some unexplained density in my crystal structure that I think is a ligand. And so we spent a lot of time trying, I'll tell you what we did, and I'll come out ahead of time saying, we have not successfully identified things in those cases. But the first thing, or amongst the things we do, is we try intact mass electrospray that I mentioned, right? So if you have a ligand and it is, it's at high stoichiometry, right? And that's important, and it's maybe hard for a given researcher to keep it at high stoichiometry if they don't know what it is. But if you purify it fresh and you think it's at high stoichiometry, by high, I mean, let's say 20% or more, right? Then we can spray it and calculate the mass, right? And then we can spray it in a way that would knock off whatever was there and calculate the mass. And then we could get a mass difference. Now, the masses we're going to measure are going to be not at super high precision, as I mentioned, because they're just so big. But in theory, then that would allow us to tell you the mass of whatever was bound to, 1 or 2 Daltons. And so if you were at the stage where you sort of said, well, look, I don't know, I have this protein, I'm working with it, I think it might bind this class of protein or small molecules. And so you have a candidate list, and we could give you some information about how to narrow that candidate list, right? That might work. The other thing you could try doing if you're purifying enough of this protein, right, is then denature it in some fashion, or just do something to release the ligand. And then we could do a more metabolomics analysis and say, well, what do we see when you treat the protein, the purified protein sample, and theoretically release whatever was there. And, that approach, I mean, we've done things like that. If you want to go that route, you might want to contact some of the other, the quote metabolomics centers around the state that do, whatever. Metabolomics is in two camps, right? There's the targeted analysis, and then there's untargeted metabolomics, which generally involve looking at large numbers of species, but also involve looking at smaller numbers of species and actually identifying them. So some combination of those two approaches is what I would suggest. And again, the caveat is we've tried them in multiple systems and haven't been successful. And it's, not clear to what extent, well, maybe they just had something that was super low stoichiometry. And so, we never saw it in the intact mass analysis, and we never saw it when they released it because there just wasn't much of it.
All right, anyone else have a question? If not, thank you, John. This has been a really great discussion, a really great presentation.
Yeah, glad I could inform people. Yeah, and like I said multiple times, just send me an email if you have any follow-up questions. I'd be glad to chat.
Okay, great. Thank you so much.
All right, take care.